Hello and welcome to Nikolai's Genetics Lessons, and today's first problem is scientists have discovered how to put together a bacteriophage with the protein code of the phage T2 and the DNA of the phage T4. If this composite phage were allowed to infect a bacterium, the phages produced in the host cell would have, and you have to choose one of these answers. By the way, this is not just theoretical question, this is practical question. And scientists conduct such experiment about a century ago. At that time, they still was not sure whether proteins is the source of the heredity information or DNA. And most of the scientists were thinking that it is proteins because DNA was too simple with only 4 bases. So such experiment was designed, and of course, now you know that DNA is a source of the heredity information. And if we use bacteriophage is a virus that infects bacteria. So if we use bacteriophage with DNA from T4 virus, of course all the progeny will have DNA of the T4 virus and protein codes also of the T4 virus. So the correct answer is D. And next question, which of the following is not needed for DNA replication? Imagine that this is DNA, double-stranded DNA, and we see here new strands of the DNA would be formed. In one direction it's going to be continuous, and in the other direction it's going to be discontinuous. But we need ribosome when we'll have messenger RNA, large subunit, small subunit, and protein chain would be formed. But we don't need ribosome in order to replicate DNA, only in order to make a protein chain. So our choice is going to be answer A. We don't need ribosomes during DNA replication. We need ribosomes only during messenger RNA translation into protein chain. And this is all for today. Thank you for your attention and see you in the next video. Goodbye.